Hello all, in this video we are going to see about CSIR UGC exam with respect to life sciences subject. We have 13 units which include molecules and their interactions relevant to biology, that is unit 1. It speaks about biochemistry. Cellular organization, cell communication and cell signaling speaks about cell biology. Fundamental processes speaks about molecular biology. And also we have developmental biology, system physiology of plants and animals, inheritance biology, that is genetics. We have diversity of life forms, ecological principles, evolution and behavior, applied biology and methods in biology. In this video we are going to see about the major highlights of syllabus issued by CSIR NET examination. In case of biochemistry, a candidate should learn, biochemistry is a must learn unit when it comes to CSIR NET exam preparation. We will be learning about structure of atoms, molecules and chemical bonds like covalent bonds, hydrophobic interaction, van der Waals forces, electrostatic interaction among various biomolecules. Then we will learn about structure and functions of each biomolecule. Biomolecule in the sense it includes carbohydrates, lipids, proteins, nucleic acids and vitamins that exist in the biological system. So we will learn about composition, structure and functions of each biomolecule. Then about stabilizing interactions. That's what I said. Van der Waals forces, electrostatic interaction, hydrogen bonding, hydroscopic interaction, it's all about that. Then about pH, buffer, kinetics, thermodynamics, how each biomolecule react under this pH, under a change in buffer, under a change in reaction kinetics, under various thermodynamical conditions. Next is about bioenergetics. In case of bioenergetics, we will learn about the basic processes like glycolysis, Krebs cycle, pentose phosphate pathway, and electron transport chain, that is HMP shunt or pentose phosphate pathway, all about that. Next about the principles of catalysis, that is enzymology. We will learn about enzymology, isoenzymes, abzymes, Michaelis-Menten equation, Line-Weaver-Burke plot and their application, how they apply to the case and the question given in CSIR exam. Then about protein metabolism, protein structure, protein function, the famous Ramachandran plot, what is motif, domains and foldings. Next about nucleic acids, A, B, Z form, tRNA, miRNA, siRNA, their conformations. Next about the stability of proteins, there's various solubility conditions at various temperature, at various pH, at their absorbance at spectrophotometer, all these we will be studying. Next about the metabolisms of these biomolecules. It includes carbohydrates, lipids, amino acids, nucleotides and vitamins. This is a must learn unit of biochemistry in case of CSIR exam. In case if you are a person from biochemistry background or from any other background, this is a must learn unit for everyone because biochemistry is the mother of life science. Anyone who start studying life science should learn biochemistry. Next thing is about cellular organization or cell biology. In case of cell biology, we will be learning about cell structure and function, how the various molecules outside the cell comes inside and the molecule inside the cell goes out, that is membrane transport, the properties of the cell membrane, that is the lipid bilayer membrane, and their interactions with the surrounding, how the protein sorting occur among each organelle inside the cell, how the protein manufactured in one organelle is being transported to other organelle. This thing we will be studying. Various pumps, various mechanism of protein sorting, electrical property, intracellular transport, all this. Next about structural organization and the functions of intracellular organelles, like that what I said, what a cell have. Cell may have, plant cell have cell wall, nucleus, about mitochondria, about Golgi bodies, lysosomes, endoplasmic reticulum, cytoskeleton, all these we will be learning. Next, this thing forms the basis of molecular biology. So this is a must learn thing for anyone. Most important aspects in exam point of view, even operon concepts, this repetitive DNA concept, chromatin concept, transposon concept. This is a must learn part for anyone who prepare for this exam. See, whole syllabus should be read by everyone who prepare for this exam, to have an overall idea about the exam, at least once, to have overall idea about CSIR expectation. But this must learn thing forms the basis for any student who prepare for this exam. So you are not supposed to skip this. Once you skip, you lack in a candidate lack the foundation. So a candidate feel difficult to understand other coming units. Next about cell division, cell cycle, microbial physiology, about the growth of microorganism, their characteristic may be logistic growth, they have exponential growth or what. We'll be learning this in detail. About the stress responses, cell cycle is again and again very important for anyone who learn. Mitosis, meiosis, regulation of cell cycle and their cell cycle checkpoints is again very important part. Next chapter is about fundamental processes, that speaks about molecular biology. Last chapter we learned about cell genetic organization, about operon concept, about transposons. In this thing we will be learning an extended version. We will be going in depth inside genes, how replication occurs, how recombination occurs, how transcription occurs, how translation occurs. We will be learning about this central dogma theory. We will be learning all processes like replication, transcription, translation in detail in case of both prokaryotes and eukaryotes. Next we will be studying about protein synthesis. It is about translation process, how the process occurs, what are the regulation, what are the key factors involved, transcription factors involved in transcription, translation factors involved in translation, what are the factors, what are their functions, how are they regulated, who are the inhibitors, when it is inhibited, how it is inhibited. We have various benefits in manufacturing antibiotics based on these processes, how it benefits us, how the control of gene expression and transcription and translation level occurs in case of both eukaryotic and prokaryotic gene expression. All these things we will be learning in this chapter. This is the overall idea of this chapter. It gives a comprehensive learning about in-depth genetics, that is in-depth advanced learning about cells in molecular level. Next, about cell communication and cell signaling, where we learn about how a cell interact with another cell. This unit will be studying about this cell-cell interaction. What are the secondary messengers involved, like cyclic AMP? We will be dealing with cyclic AMP, cyclic GMP. What are the duty of protein kinases, various type of protein kinases, various type of phospholipases, how are they involved, how they regulate our body function, various signaling pathway in case of plants, in case of microbes, in case of humans, we'll be studying. Apart from that, also be dealing with cellular communication, that is inside an organism, one cell is here. Let's assume one cell is here, another cell is here. One cell has to maintain a compatible and most positive interaction with the other cell to have a hold over it. Only then another cell can form, and by forming this we form an organ that result in organ system, that result in organism. So this is very, very important. We will be dealing with cytoskeleton complexity here, how various proteins that help in attaching one cell to another, about extracellular matrix, about integrins, about neurotransmission and regulation. We will be also dealing in neural system and chemical coordination in human physiology as well. Various adhesion molecule, how a cell attaches with extracellular matrix, how a cell attaches with basal membrane, how a cell attaches with other cell, how a cell allow its molecule to pass to the other cell, whether it have a gap junction, whether it have a tight junction. We will be dealing with this in this chapter in cellular communication. We also have an host parasite interaction. How a host may be a plant, may be a human, it may be an animal, how it interact with the pathogen when it comes inside it. That we will be learning. Next, about cancer. Most researched topic the present day is cancer. This is also a basic topic, so one should not skip it. Dealing with oncogenes. What is oncogenes? If we are learning from basic, what are oncogenes? What are proto-oncogenes? How cancer is developed? What are the signal mechanism? How to treat cancer? What are the mechanisms we should learn to treat cancer? What are the things that are found so far in cancer treatment? So to understand this, we should have a basis from cell cycle checkpoint in unit 2. We should learn about apoptosis, regulation of apoptosis. We will also learn about apoptosis in unit 5, but apoptosis forms the basis of cancer studies. Next, we will learn about immunology as well. In immunology, we will be learning with from the basis. What is immunity? What are the types of immunity? What are the cells involved in immune responses? About various molecules, antigen processing and presentation, activation, humoral response, cell mediated immune response, primary and secondary responses, autoimmunity, hypersensitivity, and about infections like TB, malaria, virus, AIDS. We will be dealing all with this in this unit. Next, about developmental biology. So one should learn developmental biology. We should know about the basic terminology. Terminology is important while learning developmental biology. What is stem cell? What is totipotent stem cell? What is totipotent cell? What is the difference between embryonic stem cell and the adult stem cell? What is pluripotency? What is totipotency? What is multipotency? About cytoplasmic determinants, about specifications, about morphogenic gradient, about determinations, competence, basic terminology is much required to go in depth of this unit. Next, in developmental biology, we will deal about gametogenesis, morphogenesis of various organisms like amphibians, chick, C. elegans, plants, organogenesis, bulbal formation in C. elegans, very important part, regeneration, metamorphosis, post-embryonic development, tetrapod lymph development, islands induction. In case of plants, we have transition to flowering and floral meristem and floral development. SAM, that is shoot apical meristem. These are the things that we should learn from developmental biology. Sixth unit, plant physiology. It is an extension of biochemistry. We will deal about various cycles: citric acid cycle, Krebs cycle, C3 cycle, C4 cycle, C2 cycle, TCA cycle. So, if a person is strong in biochemistry, it becomes very easy to handle this chapter on plant physiology. About nitrogen metabolism, nitrogen cycle, nitrogen fixation and biosynthesis. About plant hormones, phytohormones like auxin, gibberellin, cytokinin, ethylene, abscisic acid, brassicosteroid, jasmonic acid, and their signaling pathway, their biosynthesis, their function, their precursor, and how they help plants in growth and improvement. We'll be learning all these in plant hormones. Again, a very important part. Next, about sensory photobiology. In sensory photobiology, we will deal about cryptochrome, photoperiodism, phytochrome, biological clock, circadian rhythm. All these things are very, very important. Next, about how the plant transports by using xylem and phloem. How photoassimilation takes place. Mechanisms involved in transportation inside plants. Next, about secondary metabolites. Various secondary metabolites we know. We have terpenes. We have alkaloid, phenol, flavonoid, so we will be dealing all with these and their help in plants for their improvement. The secondary metabolites are also of economic importance. They are also of medical importance to humans. They also sometimes protect plants. They help in the growth of plants. Next thing is about stress physiology. Plants are exposed to various conditions: temperature, humidity, salinity, rainfall pattern, may exposed to drought condition, may exposed to logging, that is water logging, biotic and other abiotic stress of various pathogens interactions, various infections in plants, how they handle. That is all about stress physiology. Next up is system physiology, where we will learn about cardiovascular system, nervous system and neuronal coordination, endocrine system about various hormones, pituitary hormone, thyroid hormone, adrenal gland hormone, gonadal hormone, that is sex hormones, exocrine, both duct and ductless glands hormones and their functions, about excretory system, about sense organs, about blood and their circulation, various blood components, instruments like ECG, instruments like EEG, central peripheral and autonomic nervous system, waste elimination, micturition, mechanism of urine formation, thermal regulation in humans, that is various abiotic regulation in humans, abiotic stress handling in humans, digestive system, how absorption, assimilation takes place, what are the various organs involved, what are the enzymes involved and their precursors, and how it is absorbed, where it is absorbed, what are the channels involved in absorption, how the components are broken down. All these things will be learning in this unit of human physiology, that is system physiology in animals. Next, about genetics. In genetics, we will learn about Mendelian principle, Mendel's law of segregation, law of independent assortment, law of dominance. All three criteria based from the scratch, from basic. Then about the concepts of gene, multiple alleles, allelic interaction. What epistasis is about the deviations of Mendelian principle, that is codominance, incomplete dominance, genomic imprinting, crossing over, sex influence character, sex limited character, sex linked character, linkage mapping, tetrad analysis, molecular markers, extrachromosomal inheritance, position effect, then about maternal inheritance, maternal effect, interrupted mating, microbial genetics, about transformation, transduction, conjugation, sexduction, mapping genes, interrupted mating, transformation, conjugation and transduction. About human genetic disorders, how we calculate by using pedigree chart of the particular family. If the genetic disease that is inherited, we can easily trace from pedigree chart. Lot score, karyogram, genetic disorders, quantitative genetics. Numerical questions will be asked from this. We will be applying Mendelian principles to solve this, about various types of mutations. Lethal, conditional, gain of function mutation, loss of function mutation, somatic mutation, all those things. Then about structural and numerical alteration, that is chromosomal aberrations, recombination, homologous, non-homologous recombination, all these things we will be studying this. Next, about diversity of life forms. Principles and methods of taxonomy, levels of structural organization, classification of plants and animals, various taxa, various phylum, all 5 kingdom classification, Whittaker 5 kingdom classification, in depth about each kingdom and their characteristics, about the health and agriculture importance, about the various conservation strategies. Next comes ecology. Environment, habitat and niche, population ecology, species interaction, community ecology, ecological succession, ecosystem ecology, biogeography, applied ecology, conservation biology. These are very general topics, but this ecosystem ecology unit remains very important, that anybody can learn, relearn, and it is self-explanatory. So one candidate should not skip this. Resource partitioning, character displacement, history of strategies, R selection, K selection, concept of meta-population, dispersal, species interactions, community ecology, ecological succession, how the succession starts, various models that supports ecosystem, basic terminologies. What is habitat? Start from scratch. What is habitat? What is niche? What is ecosystem? What is biome? What is biosphere? Start from scratch and end in species interaction, about ecological, and end in ecological succession, ecosystem ecology, that is primary production, decomposition, terrestrial ecosystem, aquatic ecosystem, about various biomes of the world. Should study with respect to India as well, various types of forest, alpine forest, tundra forest, evergreen forest, temperate forest, deciduous forest, various ecosystem, Mediterranean ecosystem, chaparral ecosystem, taiga ecosystem, tundra ecosystem, all those things we should remember, recall, recollect and revise before this exam comes. Okay. Applied ecology, about man-made pollution aspects and their tackling strategies. Next, about conservation biology, various projects, biosphere reserves, study about all biosphere reserves and all organisms that are importantly conserved in these biosphere reserves, types of biosphere reserve, how one should make biosphere reserve, biosphere and human interaction, all these things you should know. Next, about evolution, emergence of evolutionary thoughts, origin of cells, paleontology, molecular evolution. This origin of cells deals with experiments, Oparan and Halden experiment, Urey-Miller experiment. These are their basis and their ideology of explaining things, how an organism get into earth, how an organism start life in this earth. Paleontological evidences speaks about the scale of biogeography, biogeological time scale. This is bit memory based. So revise this before exam. Then about cladogram, gene duplication, divergence, convergence, coevolution, speciation, allopatry, sympatry, island biogeography, Hardy-Weinberg equilibrium, genetic drift, adaptive radiation, gene pool. This conceptual quality is very important to do problems regarding Hardy-Weinberg equilibrium. Next about kin selection, reciprocal altruism, that is behavioral ecology. This is about behavioral ecology. Social communication, social dominance, reproductive success, parental care, habitat selection, domestication, aggressive behavior, mating patterns. This is about behavioral ecology. Next about applied biology, we will be dealing with industry-based things about biosensors, bioremediation, breeding of plants including MAS, that is marker assisted selection. This is very important for any person from life science to know. This is a bit practical in nature. Pointers can come from each thing on basic criteria. So it's better to be strong in all these. Transgenic animals and plants and their diagnostic approaches, gene therapy, immunological principles like vaccine diagnosis, tissue culture, biosource and biodiversity, biosensors, bioremediation, phytoremediation, the application aspects of transgenics, all these things. This is a very, very important topic. Methods in biology. This unit has a correlation with all those 12 units we have seen so far. This is the unit that explains about the real-time application aspects of all those 12 units. Part A speaks about the application-oriented things of biotechnology. We will learn about RAPD, RFLP, about various biochemistry aspects, sequencing, protein sequencing, carbohydrate sequencing, about gel electrophoresis, 2D gel, SDS gel, isoelectric focusing, about DNA sequencing, about this chain termination method, with this microarray chips, RACE, RAPD, RFLP, all these things. Next about the application-oriented topic of immunology, about various biophysical techniques like spectroscopy, NMR spectroscopy, UV spectroscopy, IR spectroscopy, X-ray diffraction spectroscopy, ESR spectroscopy, surface plasma resonance spectroscopy, all these things. Next about various statistical methods to prove our research studies. We use mean, standard deviation, ANOVA, correlation, regression, chi-square test, t-test, binomial distribution, Poisson distribution, about the various measures of central tendency. Next about radio labeling techniques, about various microscope, bright field microscope, dark microscope, surface that is phase contrast microscope, electron microscope, scanning electron microscope, atomic microscope, tunneling microscope, transmission electron microscope, scanning electron microscope, all these things. How they are constructed, what are their purposes, how are we supposed to use it, all these things will be dealt in this. Electrophysical methods, EEG, ECG, positron electron microscope, MRI, CAT, all these things. Methods in field biology, mark capture, recapture technique, remote sensing technique, various sampling methods. All these things we will be dealing in CSIR 13th unit in CSIR NET UGC examination. Overall, I have given a gist about the syllabus regarding CSIR NET UGC Life Sciences examination. So, in further videos, we will be dealing with each unit, exam pattern, criteria, qualifications, who can appear, what are the units to be covered, then books to read, explanations of various topics from these units be getting from this channel. Thank you for watching. Have a very good day. Thank you.